Could you make me not a co-host so I don't see all these
Oh, okay.
admit requests popping into the field of view all the time? That would be
Okay. Yep. That's done. So I've removed that. Yeah.
great. Thank you very much. Okay, I have to. Sorry, I've got to pop out of this for a second and click a box on the screen, and I can now go back to my laser pointer. Okay, yeah, so great to have everybody along in a virtual sense. So this is already very strange. This would normally be in the huge lecture theatre in chemistry, in that big Howarth one-on-one lecture theatre. But hopefully the experience will be just as good or as bad as it would have been if I'd been delivering in person. So thanks for coming along, and I guess everybody's come along because they're interested in chemistry, or maybe because they're interested in medicine, and I know that's quite often an area that students who come along to this talk are thinking about doing medicine at university, which of course is a very laudable thing to want to do, but this is really all about the role that chemists play in the discovery of new medicines, because actually it's scientists, so teams involving chemists, biologists, and other scientists who actually design and discover new medicines, and that's really what we're going to be talking about tonight. So if we take a quick historical perspective on this, if we go back 5,000 years, humankind has been always looking for cures for disease. So we go back to the ancient Egyptians, and they were using simple crude extracts from herbs and plants to try and treat disease. And that position didn't really change for the next 4,000 years. And then around 1200, we see early medical schools set up. So people started to study medicine in a systematic way, trying to work out how the human body worked. But there was, of course, no medicines as such, so no real way of treating disease but beyond these sort of fairly crude plant extracts. Then 1618, so 400 years ago, we see the publication of the first London Pharmacopoeia. And that's a book that's still published today. And it lists all of the medicines that we have available at our disposal for the treatment of various conditions. And we see there the first evidence of some chemicals creeping in. So these incredibly toxic salts of arsenic, mercury, and lead. I've no idea what they did with lead, but it may surprise you to learn that arsenic and mercury were in fact used to treat infectious diseases up until around about the 1950s, actually. But they're incredibly toxic compounds. Around about 1800, we start to see people actually purifying plant extracts and extracting pure compounds from them. And we start to see some things that we would still recognize as medicines. So quinine, which has been used for many years as a treatment for malaria. It's not a very good treatment for malaria anymore, but it was up until relatively recently used to treat malaria. Morphine, of course, a hospital painkiller, still very widely used. Strychnine, you may have heard of strychnine. It's a deadly poison, but the Victorians actually used it for its stimulant properties in very small quantities. And then atropine, so that comes from the deadly nightshade, so the name kind of gives that one away. That's another deadly poison, but it's still used today for a variety of reasons, actually. So it's used as a stimulant, as a heart stimulant, for example, and also to dilate the pupils of the eye in ophthalmology. So plants still very much have their part to play, and we'll talk a bit more about that as we go through. So if you were unlucky enough to require surgery a couple of hundred years ago, it would have been a pretty brutal process. So this is what medicine was really like back in those days, and we're not looking that far back. So start of the 19th century. So no anesthetics, no painkillers, no way to control infection. So let's imagine you had, as this poor chap has here at the top, presumably he's got some kind of injury to his leg, which has caused an infection. There's no way to treat that. So if it starts to spread around his body and it goes to full-blown blood poisoning, then he's dead. So the the simplest solution is for these burly chaps to hold him down and cut off his leg as quickly as possible. And the mark of a good surgeon was how fast he could sew off a leg. It would have been a horrendous process. You can see this chap doesn't look very pleased about it all, but it might save his life if he survives the operation. The next big danger, of course, is that the wound will get infected. So they will probably treat it, in some kind of crude way to try and stop infection setting in. But there was a good chance that a bacterial infection would set in, and he might well die from that. So yeah, medicine wasn't a particularly pleasant thing until chemists started to get involved with it. So the first chap you see at the top here is Joseph Priestley, and you might have heard of Joseph Priestley. He was an interesting guy. He was active in politics, in religion, and also in science. And his political views actually were relatively unpopular. And so he was a Yorkshireman. He actually moved down to Birmingham at one stage in his life, and this is his house in Birmingham, which in 1791 was burnt to the ground by rioters because they took offence to some comments that he'd made. So this house, he lived, I think it was Sparkbrook he lived. So these were the Birmingham riots of 1791 when his house was burnt down. But the reason he's here on this slide is that he discovered nitrous oxide. He discovered a few different gases actually, but nitrous oxide was one of them, which of course we might commonly call laughing gas, and was used as an anesthetic from the mid-19th century. So in dental anesthesia in particular, and it was used, and well, in fact, it still is used. In the dentists tend not to use it if they can avoid it, but it still can be used in dental anesthesia. So here's a chemical which would have transformed the way in which surgery could occur. Imagine having your tooth pulled out with no anesthetic versus being knocked out with laughing gas and having it pulled out, and in a relatively pain-free procedure. Another anesthetic, you may be aware of the anesthetic properties of ether, so a fairly simple chemical, but again, one that transformed surgery. So from mid-19th century onwards, so the first example of it being used in surgery was from Robert Liston in 1846. So moving on about 50 years from the previous picture where those guys were rather brutally amputating that chap's leg, about 50 years on, that would have been possible. It would have been possible to do that with anesthesia. And then a few years later, Joseph Lister, of course, started to understand the idea of bacterial infection and how it was important to keep surgical wounds scrupulously clean. So he first used another simple chemical, carbolic acid, we might call that phenol these days, as an antiseptic. So chemistry started to move medicine on really in the mid-19th century. But really where I want to pick up the story is with this chap here, William Henry Perkin. So Perkin is obviously a very old man when he's pictured in this photograph here, but he was a young man at the point at which this story begins. So Perkin was born in the East End of London. He was the son of a builder, and his parents were very keen for him to become an architect, but Perkin was actually very keen to study science. He wanted to become a chemist. So opportunities to attend university were fairly few and far between in those days. So he did the next best thing, which was to get himself a job as a laboratory assistant to a German chemist called Hoffmann, who at the time was working in London. And Hoffmann was interested in this chemical here called quinine. So quinine, we've already mentioned, is an anti-malarial. It's a natural product, and it actually occurs in the bark of the cinchona tree. So the cinchona tree is this thing here, and that is a species native to Peru. So supplies around the world were relatively limited, and Perkin's boss, Hoffmann, had the idea, could chemists actually synthesize quinine? So could they make it from scratch? Now, that was a pretty tall order. The first time an organic compound had ever been synthesized in the lab was 1828. A guy called Friedrich Wöhler made urea. And before that, there was something called the vital force theory that suggested that organic compounds could not actually be made in the laboratory. There was some kind of mysterious vital force that was responsible for producing these things in nature. So things had obviously moved on in the preceding 30 years or so, and people were starting to think, could they actually synthesize relatively complex natural products like quinine? Of course, they didn't know it was complex. They didn't actually know what it was. The only information they had on it at the time was its empirical formula. And quinine actually has quite a complex structure. So you'll see these bonds here, which are either sort of highlighted in bold to indicate that this group is coming towards you, or dashed to indicate that it's going behind the screen. So this is, if you've done stereochemistry, this is stereochemistry. These are what we call stereocenters, or maybe more loosely chiral centers. And that basically means that this molecule is very, very challenging to make. If we invert this position here, so we put this as a dashed line rather than a bold line, that's a different compound. It will have a completely different biological activity. And that's really where the challenge lies in quinine, making it with all of these stereocenters correct. And in fact, the total synthesis of quinine wouldn't be completed for about 150 years after Perkin attempted to do it. So Perkin was well ahead of his time, but with the naivety of youth. So he was working for Hoffman. He thought, well, I'll get one over on the boss. I'll make quinine. So in 1856, Perkin's 18 years old, and in his spare bedroom, he got a hold of some chemicals and he started messing about with them. He had no idea what he was trying to do, but he just sort of mixed these chemicals together, boiled them up, and of course he didn't make quinine. All he made was a big mess, really. So he made some horrible kind of black tar, and he was a bit despondent, and he started washing it out down the sink. And he found when it was diluted with water, in fact, it wasn't black. It had this beautiful purple color. So he hadn't made quinine, but what he had actually managed to stumble upon purely by accident was the world's first synthetic dye. This compound here, which was christened Movine. Now at that point, so middle of the 19th century, we had access to dyes, but they were fairly muddy kind of browns and greens and reds and so forth, not particularly pleasant colors. There were some very beautiful dyes that were available, but they were hugely, hugely expensive. So they were from natural sources, but they were very much the preserve of the wealthy. So something that could be made in large quantities and very cheaply was actually turned into a huge hit. So this was absolutely wowed Victorian society. So every woman wanted a dress dyed with Movine, and it sparked a big fashion trend. And Perkin, being a fairly enterprising young man, set up a dye factory and actually made himself extremely wealthy on the back of this. And he's sort of known as the father of the chemical industry, really, for his work in establishing dyes in this dye production worldwide, really. So he went on to make other sorts of dyes. So you're probably familiar with azo dyes. You might have even made one at school. If you haven't, then ask your teacher. They're quite easy to make. And Perkin went on to make some of these, but the Germans got very involved in the dye industry, and really Germany became one of the great world centers for productions of dyes. So, and that's really where our story moves on to next. So Perkin is left making dyes and counting his money, and the story continues really with this chap here, Paul Ehrlich. And Ehrlich was a very interesting guy. He was a chemist, he was a biologist, he was also a physician. And one day when he was in his lab, he was using some of these azo dyes to stain biological samples, looking at them under his microscope. And he noticed that the azo dyes had a very strong affinity for bacteria. So you could stain bacteria very well with azo dyes, and that wasn't true of human cells, for example. So he had this idea, well, could we actually use an azo dye as a way of targeting something toxic to a bacterium and killing it? And he came up with this idea of so-called chemotherapy. So this is the whole idea of modern medicine, that we can selectively come up with a chemical that will selectively attack a disease and not be harmful to the human. And Ehrlich actually termed this the magic bullet. So the thing that could kill the disease without harming the host. And he tried unsuccessfully to take simple azo dyes and attach toxic things to them. He didn't have a great deal of success there, but he sparked the whole idea that chemicals could be used selectively to treat disease. And as you can see here, he won the Nobel Prize in 1908 for medicine. Ehrlich didn't leave things there. He went on to, he was obviously a very enterprising chap, and he went on to found basically his own pharmaceutical company. And he was interested in treating a disease called syphilis, which it's a nasty sexually transmitted disease. It was very much the kind of HIV/AIDS of its day, and there was no treatment for it. So his work on azo dyes had sort of led him down the path of trying arsenic compounds, because arsenic is well known to be very, very toxic. And he's established a research program looking for toxic arsenic compounds that might be effective against syphilis. And he eventually hit upon this compound here, which is called salvarsan, also known as 606. And that comes from the fact that it was the 606 compound that they tested and found this one to be effective. And that just gives you a sort of a flavor of, even then, more than 100 years ago, how much chemical synthesis was involved before a successful compound was found against a disease. So they had to make 605 unsuccessful compounds before they hit upon this one. And it was revolutionary, having something that could treat syphilis. So the name salvarsan, so the arsan bit comes from the fact that it obviously arsenic, but the first bit of the name comes from the fact that it was regarded as a salvation. It was salvation from this hideous disease that disfigured and eventually drove people mad once it got to the final stages of syphilis that infected the brain. So they developed this in 1909, and this gives you a feel for what regulatory mechanisms were in place or not in place in those days. So by 1910, it was in clinical use, and Ehrlich had established this factory. You can see all these workers beavering away, filling up the vials with salvarsan. And in fact, it was used to treat syphilis until penicillin became available, and that was not until towards the end of the Second World War. Continuing on the azo dye theme, we move on to this chap here, Gerhard Domagk, and another Nobel Prize winner. So Domagk worked for one of the big German dye companies, and really in a very forward-thinking approach. They were testing their dyes to see whether they had any medicinal properties, and that was Domagk's job. So he took this red dye here, Prontosil, and he injected it into a mouse which had a bacterial infection, and the mouse got better. So the dye was obviously acting as an antibiotic, and this was an absolutely revolutionary discovery, because up to that point, so we're talking the 1920s when this discovery was made. So in the 1920s, the infant mortality rate was above 10%. So more than 10% of babies would die before they reached their first birthday. And that was because of, well, generally things that are very easily treatable now. So things that we can vaccinate against, like measles and so forth, but also simple bacterial infections that we are able to treat these days using antibiotics. In the 1920s, there were no antibiotics. So the idea of something being able to treat a bacterial infection was absolutely miraculous. So he was obviously very excited by this idea and wanted to test it on a human patient. So again, no kind of clinical protocols required. They simply went to a local hospital. They found a baby that was basically at death's door with a bacterial infection. Nothing to lose really, so they injected the baby with the red dye. So the first thing that happens, the baby went bright red, but the red color faded after a couple of days, and the baby made a complete recovery. So they demonstrated that they could actually make an antibiotic that was effective in humans. And it was only a few years later, actually, that Domack was able to save the life of his own daughter using the compound he'd discovered. So it turns out the red compound is not actually the active drug. It's what we would call these days a prodrug. So it's something the body converts into the active drug. So the body actually reduces the NN double bond into this much simpler sulfonamide. And in the 1920s and 30s, production of sulfonamides was explored very extensively. So the companies were set up and made hundreds of different sulfonamides, or so-called sulfur antibiotics. And these were the only antibiotics that we had available until penicillin became available in around about the time of the Second World War. So the sulfur drugs were by no means the perfect antibiotic. They didn't have a very broad spectrum of activity, but they were much, much better than the alternative, which was effectively nothing in those days. So here's Prontosil, the red dye in little ampules. And here, once they'd established that actually Prontosil wasn't required, just this simple sulfonamide was required. This is actually from the Second World War. This was put into the packs of soldiers in powdered form, and they were told to sprinkle it on wounds and the like to minimize infection. So moving on to penicillin, of course, that's the daddy of all antibiotics, and I'm sure you all know the story. So penicillin was discovered in 1928 by this chap, Alexander Fleming. And the story goes that he was a bit of a messy worker. He was growing some bacteria. He was looking for antibiotics, but he chanced upon it purely by accident. So he was growing some bacteria in a petri dish, and some dust got into that petri dish and mold spores with it. And around the mold growing on the petri dish, you can see the bacteria have all been killed off. So because he was looking for antibiotics, he was able to put two and two together and work out that the mold was producing some sort of antibiotic that was killing off the bacteria. And of course, the rest is history. The antibiotic that was being produced is penicillin. But Fleming was unable really to sort of scale up production. He was able to extract small quantities and identify what it was he was working with, but he was never able to actually produce any significant amount. And he was a bit frustrated by that, so he sort of published the paper and moved on to do other things. And it wasn't until about 10 years later, so just before the start of the Second World War, that these two chaps who were working in Oxford, so Florey and Chain, chanced upon Fleming's paper and thought they'd have a go at scaling up the production, because they knew all about growing up mold and extracting compounds from it. And they were able to successfully scale up the production of penicillin. So they got back in touch with Fleming, and obviously there was a big drive to scale up penicillin during the Second World War. And they'd sort of got that cracked by the end of the Second World War, really. And the three of them shared the Nobel Prize in 1945 for the joint sort of discovery and production of penicillin. So penicillin works by inhibiting bacterial cell wall biosynthesis. So the cell wall is constructed of sugar molecules, which are these spheres down here, and they're cross-linked to make a very strong cell wall by these little ties, which are peptides. And penicillin interferes with the cross-linking of the cell wall, so it weakens that cell wall and the bacterium dies. And it really was this sort of miraculous discovery that came about and was I'm sure influential in turning the tide of the war. So thanks to penicillin, he will come home, and when he gets home, there's possibly he may require a bit more penicillin, but we will move swiftly on from that. In terms of what penicillin is able to to do. It's a much broader spectrum antibiotic than the sulfonamides. And if you want to try and put a number on it, it's been estimated that it's saved at least 200 million lives since it was discovered. If we think about how the infant mortality rate has changed, and that's not just because of antibiotics, it is of course because of vaccination and general improvements in healthcare and so forth. But 100 years ago, the infant mortality rate was above 10% in the UK. These days, it is around about 4 in 1,000 children who don't make it to 1 year of age. So it really, we have transformed the treatment of infectious disease. So here's a girl who was treated with this compound in 1942. So one of the really early people treated with it. And you can see she's got some kind of horrible infection here, which had been treated with sulfonamides and unsuccessfully, but you can see after a few days of penicillin treatment, she's back to normal. So really was very much a miraculous drug. But of course, things are starting to go very rapidly in the wrong direction for antibiotics. So everybody's heard about the problems of antibiotic resistance, and penicillin. So I mean, the joke here, even penicillin won't be able to harm you. Penicillin is actually one of the least effective antibiotics that we have these days, because it's been around a long time and most bacteria have managed to build up some resistance to it. And in fact, even Fleming back in his paper in the late 20s flagged up that resistance would inevitably develop to it. And it's the consequence, as we see here, of selective pressure being applied to evolution. So of course, bacteria reproduce at an astonishing rate, and as they mutate, as they're reproducing, those bacteria who are resistant to penicillin will simply start to thrive. So the more we use the antibiotics, the more chance that bacteria will build up resistance to them. And this is a problem which of course has been hugely exacerbated by their misuse. So farmers all around the world give their animals antibiotics, not because the animals are ill, but because it simply makes them put more weight on. So they get fatter, they can sell them for more money. In many countries around the world, it's possible to go and buy antibiotics over the counter. So you're not feeling very well, you go and buy some antibiotics. Actually, you don't have a bacterial infection, you've got a viral infection, and antibiotics aren't going to do anything about that. But you just take them anyway, and that just helps to exacerbate the problem. And then paradoxically, in the developing world, they don't have the money to buy the necessary antibiotics. So they might, if you've ever had antibiotics, the one thing it always says on the label is complete the course. So, you know, people start to feel better after a few days. The tendency is just to ditch the antibiotics. But no, you have to complete the course, because the few bacteria that are still left in your system, they're the ones that the antibiotic is struggling to kill. And if you let those start to multiply, then they're the ones that are resistant to that antibiotic. So if you're in the developing world and you're short of money, maybe you don't have enough money for the full course, and that is exacerbating this problem of resistance. And as for the last point, well, globalization accelerating the spread of disease, I think we don't really need any lessons on that one at the moment. So antibiotics there are many antibiotics in addition to penicillin, but it's definitely an area which I would say is a growing health emergency. And if we don't put the lid on this pretty quickly, we're going to get to the position when we actually don't have any effective antibiotics, or have very, very few effective antibiotics. So it's really something that needs global coordination. But at the moment, obviously, our attentions are focused elsewhere. So let's have a look at what modern medicinal chemistry is all about. So it's all focused really on finding this thing in the blue box, the so-called lead compound. And as the name would suggest, that is not itself a medicine, but it is something that should be able to lead on to a medicine pretty quickly. And these days, lead compounds are discovered through a process called high throughput screening. So that's the idea of pharmaceutical companies being able to screen thousands, if not millions, of compounds through largely an automated process and identify things that are effective against disease targets. So of course they need lots and lots of compounds, and they get them from what they rather grandly term their compound libraries, which are just collections of compounds that they have. And they're constantly adding to these. So chemists in the lab are synthesizing new compounds and they add those to the library. And people are going out and finding new things in nature and adding those to the library. And even if they don't necessarily become a medicine, they will provide a good lead that might lead on to a medicine. So they for whatever reasons, they build up collections of compounds looking at one disease, but when the next disease target comes along, they would expose that to their compound library and see if there are any active compounds in there which might affect that disease target. In the sort of post-human genome era, so we now have human genome data, and we can use that in two ways. So we can use it to identify disease-associated proteins, and they can be fed into the screens. So these screens, we're not talking about animal screens at this point. We're just simply looking at a simple readout as to whether a compound maybe inhibits an enzyme or something like that, whatever the particular target may be. So that's one thing we can do. Once we've identified from genome data what the associated protein is, we can use that in the screen. But we can also get a structure of that protein, maybe using a technique like NMR or X-ray crystallography. And once we've got that protein structure, we can use that to try and design a molecule to block its function. So some pharmaceutical companies specialize in this type of approach. So they get the proteins, they get the structures of them, and then they design molecules to try and block the function. And either method will then lead to this thing in blue, the lead compound. To get from the lead compound to the drug is a hugely time-consuming and expensive process. So we'll be talking tens, hundreds, thousands of millions of pounds to get maybe from a lead to a drug. And it will involve many years of work. So you're probably looking 10 to 15 years of work, depending on what the drug actually, what the target is. So what do chemists do during that period? Well, the lead compound will be far from perfect. It will clearly have some activity against the disease target, or you wouldn't have picked it. But it's probably not going to be great to put into patients. It might not be as active as you would like it to be. There could be side effects and various things that you need to engineer out of it. So what chemists will do is they will synthesize analogs of that compound. They will put them back into the screens, and they will see whether that's better or worse. And on the basis of the result of that screen, they will synthesize more analogs, and they'll keep going around this route. And they might make several thousand compounds in an attempt to optimize that lead. They'll then feed that lead into the clinical testing program. So they'll come out with a clinical candidate, probably 2 or 3 clinical candidates, so there's a backup, and they'll put them into clinical trials. We'll talk a bit more about that later. And if that's looking promising, then at some stage, chemists will come back in and start developing a process to scale that up to large-scale production. So for clinical trials, you might need a few hundred grams, maybe a few kilos at most, and you could make that kind of quantity in a lab without any problem. If you're going to make a big blockbuster drug, you might need ton quantities of it. Then you're probably not going to make that in the same way. So chemists will come in who specialize in the process of scaling that up to a large scale. And eventually, at some point, you'll get that drug onto market. So let's have a look at a few examples of drugs that have made it through to the market. So we started talking about quinine. The challenge of quinine, of course, is its complexity. It's a very difficult molecule to try and synthesize. The reference there for those of you who've done stereochemistry, but it's possible sometimes to take a lead compound from nature like this and actually to simplify it, to strip away much of that complexity and still maintain function. So in the 1940s, this compound chloroquine was discovered, and that's actually a very easy compound to make, 2 or 3 steps in the lab. It's much simpler than quinine, but it's just as effective at treating malaria. So sometimes it's possible to take a complex target, complex lead compound, and simplify it and retain activity. Other times it's not possible. So this is the story of Taxol, which is currently the most successful treatment that we have for breast and ovarian cancer. And it comes from the bark of the Pacific yew tree, Taxus brevifolia. And it was discovered in the 1960s, put into human trials in the 80s, and eventually marketed in the 90s. And it's a very complex molecule. It's very, very effective, as you can see from the data up here, at treating those cancers, but it's a problem of supply and demand. There's a big demand for it, and the supply is minimal. So these trees grow only on the west coast of America. They take about 100 years to reach maturity, and you've got to chop a lot of them down to extract enough Taxol to treat just a single patient. So what can we do? What can the chemists now do to try and maintain supply? So unfortunately, there's no practical route to make Taxol. It's a phenomenally complex molecule. Some research groups have synthesized it. It's nothing that you could ever do in any kind of practical way. So it's many, many steps which are very difficult to carry out, and you can really only make milligram quantities. So they synthesized it to prove they could make it, to verify its structure and so forth, but that would never provide a route to sufficient quantities to treat patients. So the next approach, what about stripping away some of the complexity and going for a much simpler molecule? Well, as people were synthesizing it, they tried making analogs en route and found that they had no activity whatsoever. So unfortunately, these cancers are challenging targets, and they will not accept simpler analogs as therapeutic molecules. So chemists started casting their eye around for alternatives, and Taxol, as I mentioned, comes from the bark of the Pacific yew tree. But it turns out that all yew trees, including ones that you might have growing in your garden, contain this compound here, diacetylbaccatin. It's in the needles of all yew trees. And the needles of the trees, of course, are a sustainable resource. You can clip the trees and extract this compound from the needles quite easily, and it's relatively plentiful. And if you compare this with this, you'll see that this is actually the right-hand half, and by far the more complex half of Taxol. The left-hand half is this. Well, that's a pretty simple molecule. We can make that in the lab in a couple of steps, and the two are joined together in the form of an ester, so very easy to make in the lab. So Cancer Research UK, for example, will take yew tree clippings from anybody who can provide them in quantity. So the National Trust, for example, will provide tons of yew tree clippings to CRC UK, and they will extract this compound and marry it up with this compound in the lab to make large quantities of Taxol. So touching there on this area of structure-activity relationships. So sometimes it's possible to simplify a molecule and maintain activity. Other times it's not. So which bits of the molecule are important? So medicinal chemists will set out to establish what they call structure-activity relationships. And there's a nice story if we look at morphine. So morphine comes from the opium poppy. It's been used for thousands of years as a painkiller. But it has its side effects, one being it's addictive, and the other, another one being that it's very easy to overdose on it. So really people would like to find safer painkillers that are less addictive. So they set about making analogs and seeing how that affected the activity. So there we've got morphine, and there we've got codeine, which is a simple methyl derivative, and it's much less active, as you can see. In test tubes, it's only about 0.1% activity. in the body, actually, it's about 20% as active, which indicates that the body is actually taking this as a prodrug, chopping off that methyl group and making morphine in vivo. But codeine is a useful painkiller. You can buy it over the counter, and it just gives you that very low dose of morphine. It's possible to make other derivatives. So people explored making esters and found they were more active. So we've got that one, and we've got this ester here, which is the double acetate, otherwise known as diamorphine, which is very widely used as a hospital painkiller. So if you're ever unlucky enough to be in hospital and require strong pain relief, they will probably give you diamorphine. And this compound was first synthesized around about 150 years ago. It was thought, I don't quite know why, but originally when it was discovered, it was thought to be non-addictive, and it was used actually in cough mixture for children. I can assure you it's no longer in cough mixture for children, because the other name for this compound is heroin. And it got that name because of the kind of heroic feeling that people got when they took it. Well, surprise, surprise. So it's a very potent painkiller. It is the doctors would always call it diamorphine. They would never say they were going to give you heroin, but it is actually the same stuff. So lots of other analogs have been made, and you can see the bits of so I've tried to highlight the bits of the molecule that are important. So for example, pethidine, very commonly used fast-acting painkiller in hospitals. So I've highlighted the bits of the molecule which are similar. You can see we've got the benzene ring, we've got this sort of bridge to the nitrogen here, but we can strip away a lot of the molecule and still retain activity. Methadone, I'm sure everybody's heard of that. That's the stuff they give heroin addicts to try and wean them off heroin. You can see it bears very little resemblance to morphine, but it's still got this ring, it's still got this chain going to this nitrogen. So this is what structure-activity relationships is all about, really, trying to work out which bits of the molecule are important, and in the process uncovering interesting changes in activity as you start to change different bits of the molecule. And it's possible to push this in a completely ridiculous direction the other way. So this compound etophine was discovered in the 50s, and it's actually much, much more potent than morphine, so much so that you could never use it on a human. It's used in these tranquilizer darts for elephants and the like. So if the vet wants to do an operation on an elephant, they've got to tranquilize it, they'll shoot it with a dart that contains etophine. And it's so potent it will knock out an elephant. And when you want to bring the elephant round, well, the antidote exists as well. So this compound was discovered to be an antidote. So again, there's morphine. We've modified the structure quite a lot, but this actually has completely the opposite effect. So it completely blocks the activity of morphine. So a compound like morphine will be called an agonist. So it's something that binds to a receptor in the body and exerts an effect. And a compound that blocks that is known as an antagonist. So diprenorphine is a antagonist for etophine. So it'll bring the elephant back round again after the operation. This compound here, naltrexone, is what they would give perhaps an addict who'd taken an overdose of heroin. So if somebody's rushed into hospital having taken an overdose, they can give them a shot of naltrexone, and it should counteract the effect of the heroin. So agonists and antagonists are useful terms that we'll come across again. So we've talked a lot about sort of people discovering medicines from plants, really, and I wouldn't want to leave you with the impression that that's where we get the majority of our medicines. These days, it's far from that. And actually, we went through a sort of a golden age in the 50s and 60s of people designing medicines by a purely logical approach. So this guy, James Black, and another Nobel Prize winner, so he was working for ICI in the day. So you probably never heard of ICI. It became Zeneca, then AstraZeneca, then AZ. So one of the biggest pharmaceutical companies still around. And they were looking at receptors in the body for adrenaline, so the so-called adrenergic receptors. And so receptors are molecules to which adrenaline will bind. So we're talking about proteins in the body. And there are all sorts of different proteins that will bind adrenaline, and they will have different effects. And so these are termed adrenergic receptors, and they're given names alpha and beta and so forth. And depending on whether adrenaline binds to, for example, a beta-1 receptor in the heart, it will increase the heart rate. If it binds to a beta-2 receptor in the airways, it will cause the airway to dilate. So potentially very powerful physiological effects that can be used to treat some very common conditions. So there's adrenaline, and it's never that easy these days, but back in those days they were able just to make some simple analogs. So this one, propranolol, is a beta antagonist or a beta blocker. You might have heard of beta blockers. So people sometimes get them for things like conditions like anxiety, but also they're used to treat generally conditions of the heart and circulation, like angina and high blood pressure and so forth. And a beta-2 agonist, so we mentioned here that beta-2, things that bind to the beta-2 receptor in the airways cause it to dilate. So an agonist is something that will bring that about. And if anybody here has asthma, they will have probably used this stuff. So this is salbutamol, which is in the Ventolin inhalers. So invented by this chap back in the 60s, James Black. So that's really using our understanding of, so chemists working together with biologists and physiologists to use our understanding of what the body is doing to try and design a molecule to manipulate one of those pathways. I'm going to skip over the HIV slides and move on to this story. So this is this now brings us sort of into really the more modern approach to medicine. And this is a compound sildenafil, which was discovered through high throughput screening. So this was a program being led by, in fact, a Birmingham graduate, a guy who was knighted for his work on this, Sir Simon Campbell. And Pfizer were looking at an enzyme called PDE5, which is important in angina. So the angina are these chest pains that you get if there's restricted blood flow to the heart. And so they were looking for something that would treat angina. And they got this this sort of hit from their high throughput screening, and they worked it up after rounds and rounds of work. So this would be many years of work, making thousands of analogs. They eventually identified this compound sildenafil, and they put it into test tube models and found it was very, very active against PDE5 in test tubes. But they found in the human body, it had a very disappointing effect. So that, of course, is just the kind of cut and thrust of medicinal chemistry, really. You get a compound which is looking hugely promising, it gets into clinical trials, and it just completely tanks. And that is hundreds of millions of pounds worth of work down the tubes. It turned out actually not to be such a bad story, though, because they discovered during the clinical trials, whilst it wasn't active against angina, it had a rather interesting side effect that the young healthy volunteers were reporting. And to cut a long story short, this compound was marketed as Viagra, and is probably the biggest selling drug in the history of pharmaceutical industry. So sometimes chance discoveries can lead to very lucrative products. If we think about the whole process of development and testing, so I've said it's a very expensive business, and that's really because, first of all, it takes a long time, but also because a lot of compounds don't make it. So we saw sildenafil, fortunately for Pfizer, did make it in a slightly different way than they had planned, but lots of compounds actually bomb out when they get into clinical trials. And all of the development work that's happened up to that point is completely wasted, and that needs to be covered in the pharmaceutical company's budget. So for every drug that makes it to the market, many don't, and the overall cost of a single drug that makes it to the market is estimated around about, I mean, you could really put in any figure you like here, because it depends. It's sort of getting worse and worse every year as we go for harder and harder targets. But we're looking at several billions it costs to develop a new drug these days. So you might get your hit compound quite quickly from a screen. But to work that up, you might make 5,000 to 10,000 analogs in the lab. So that's a lot of chemists working away for many years to make compounds and putting them through rounds of testing with the biologists and so forth. And then once you've got something which is looking really promising, it's got to go through this rigorous approvals procedure. And we saw earlier the fact that Ehrlich, for example, could discover a compound, and boom, a year later it was on the market and being used to treat patients. These days it's completely different. So there's a very rigorous approval procedure, and one of the things that strongly influenced that was a story you may have heard of this. If not, if you're doing A-level chemistry, I'm sure you will hear about this story, and that's the story of thalidomide. So thalidomide was a drug that was marketed in the 1950s and 60s as a cure for morning sickness. And it was a successful drug in treating morning sickness. And to be fair to the company, they had actually tested it quite thoroughly, and they'd tested it even on pregnant women. But they hadn't tested it on pregnant women in the very early stages of pregnancy. And it turns out that thalidomide is incredibly toxic to the fetus in the early stages of pregnancy. So what happened is that the many babies were born missing limbs and other perhaps internal organs as well. So the testing had been insufficiently rigorous. And it was only a problem for literally something like 1 week of the pregnancy, but they'd missed that. The problem is, again, if you've done chirality, so the idea of objects, molecules existing as object and mirror image, then the problem is actually one of chirality. So this enantiomer, this mirror image form, is the one that cures morning sickness, whereas this compound, which is the exact mirror image form, if we put a mirror down the middle here, that will be what will be reflected exactly across. This enantiomer, as we call it, is the one that causes birth defects. So these days, whenever a compound like this, a so-called chiral compound, is being used in medicine, we have to test both of the enantiomeric forms separately and together to see if there's any problems. So taking a drug, for example, which is, you may have heard of Prozac, a very common antidepressant. So this is a chiral compound. If we separate the enantiomers, we found one of them is effective against migraine, whereas the other one is completely ineffective against migraine. The mixture of the two together is actually what is used as the antidepressant, but it's important to test both separately to see if there are any potential problems. So I've sort of alluded to clinical trials, and these are a very time-consuming part of the drug discovery process. It probably takes something like 10 years to get a compound through clinical trials, so 8 to 10 years. It starts off with phase 1, which is just a small group of healthy volunteers. So students quite often volunteer for clinical trials because they're they're not disposable, because it's very well paid, and they just kind of need to lounge around watching TV, and they're just monitored. So it's quite easy work, and if the students survive that stage, of course they survive that stage, then the company moves on to phase 2 clinical trials, which will actually start to use diseased patients. So a small group of patients with the disease, looking to see whether it's actually efficacious. So the first group, phase 1 trials, are simply looking to see whether there are any kind of side effects, what reasonable dosage would be, but then they move into seeing how effective is it in treating patients. And then finally, if that's looking promising, they do a longer term study with a much bigger group of patients. And so we're looking at a minimum of sort of 8 years really to get through this process. And many drugs will bomb out. Some will bomb out after phase 1 trials, more commonly after phase 2 trials, when it's found to be not as effective perhaps as originally planned. And perhaps some will not make it through the final stage. So very frustratingly, huge amount of money has been invested by this point, and they might bomb out in the phase 3 trials. If things are looking promising during those trials, then chemists will, the process chemists will come in. As I mentioned earlier, they will scale that process up. So going from lab scale to plant scale, and that will involve very different type of chemistry and a very interesting type of work, really, very different challenges from working in a lab. Very important, of course, to protect your discovery. So I said it costs something like 4 billion to develop a drug, and the revenues. So take a drug like Viagra. Whilst that was still on patent, it was grossing several billion every year for Pfizer. So this is how they make their money back again, but it costs them billions in development costs. So you need to protect that investment by very tightly tying new discovery down with a patent. So chemists will work with the lawyers to write a patent which is hopefully watertight and will stop anybody else stealing that discovery. So there's lots of different things a chemist can do in the drug discovery process. So for example, you could become a natural products chemist, going out into the wilds, discovering new plants, extracting natural products from them, elucidating their structure. Maybe you could go into protein chemistry, elucidating the structure of proteins as targets. But probably the majority of chemists who go into the pharmaceutical industry go into synthesis, and that really is the sort of bread and butter of the pharmaceutical industry. So medicinal chemists will make compounds, but will use their knowledge of how that compound might interact with the body to then design an analog and run a program of drug discovery in that way. These days, that's often guided by computer modeling. So some chemists specialize in sort of the computational aspect of computing. So they will look at how a compound might dock into a protein target and maybe use that knowledge to try and guide the synthetic work. Other chemists like to go into analytical work. Very important aspect, particularly in the drug development process, identifying toxic impurities that might be causing side effects. And as I've already mentioned, process development, that's a very different, also very interesting area of chemistry, looking at scaling up from a few grams up to perhaps ton scale. So the chemist very much is at the center of the operation, interacting with all of these different people to discover the medicines and to protect them. And the chemist really is the heart of the whole operation. And I like to think that if it wasn't for chemists, really medicine would still look a little bit like this. So thanks very much for your attention. Happy to answer any questions if you've posted any questions in the chat, but I will stop screen sharing there if I can work out how to do that. I'm going to go back to the arrow, and yeah.
Excellent. Thank you very much, John, for that. In fact, while Jill just says her thanks, if I could just remind people, if you would like to put a question in the chat, there will be opportunity. I'll read a few out afterwards. So, sorry, Jill, over to you.
Thank you. Thank you. I'd just like to thank everybody who's here for attending the first West Midlands Chemistry Teachers Centre sixth form Zoom lecture, a different experience for us all. So thank you for coming. I'd like to, on your behalf, thank John for this most informative and interesting history of medicine. So thank you very much. And I'd also like to thank Mark Reed, who's also from the University of Birmingham, like John, for his work in setting up these online lectures for us all. The next lecture of the series is in 2 weeks' time, and John's mentioned chirality, and that will be discussing and looking at chirality. So if you've done some work on it, if you're year 2, or if you're year 1 and would like to find out more, the next one is in 2 weeks' time, and it's Simon Cotton who's leading that. So thank you very much, John. Most informative.
That's a pleasure. I can see lots of messages of thanks flying into the chat, so thank you very much, everybody. You're very welcome.
Indeed. If I can just ask, John, there was a very interesting question from one of the students here, if I may. The question is, going forward, could it be better to identify the properties and possible applications of any molecule discovered and put this in some sort of database for use in targeting diseases whenever needed, rather than identifying a target disease first and only then looking for candidate molecules? So could the first method of drug development, if done before the current pandemic, could it have been useful in aiding COVID-19 drug development?
I think that's a really interesting question, and I think you probably put your finger on what is the future of drug discovery. Definitely. So, medicinal chemists these days think a lot about what is termed chemical space, which is really a way of thinking about how you might map space in 3 dimensions. If you take molecules, you plop them onto a 3-dimensional grid. Have they got, for example, a polar group in one region, a non-polar group in another, and you can map compounds in that sort of way. So what chemists are sort of currently thinking about is how to design libraries of compounds that very effectively cover a large amount of conformational space, or chemical space rather, without actually having billions and billions of compounds. So can you come up with a sort of a minimal set of compounds that will have the right kind of polar functional groups that we know work really well in medicines, covering different areas of chemical space. And then once you've identified a particular target, you know what sort of molecule you need to bind to that target, then you could potentially pluck it out of that virtual library. And as a computational chemist yourself, Dr. Reed, that is something you might that might chime with you. But yeah, definitely for sure. That's I think the idea of having a compound on the shelf that could be used to treat a disease that we don't yet know, I think is probably a bit pushing it a bit far. But certainly we could have very well developed libraries. So as soon as we identify the target, we could say, yes, the kind of thing we need to go for is that, and we've got those libraries of compounds, and that would really accelerate the screening process. So yeah, great question.
Excellent, thank you. There's one interesting one here. Is there any potential for the use of venom in medicine, in your opinion?
Probably, in my opinion, would be yes. I mean, certainly, I think so. Venoms tend to be quite complex sort of peptides. I think I don't know a vast amount about venoms, but yeah, I think for sure all natural products can yield interesting leads for medicinal chemistry.
I can see we've got another question from Dennis YM there as well, Dr. Reed.
Oh yes, sorry, that was just on my list. Yep. So yeah, this question: do the computer models get any better? I might answer that one for you, John. For example, originally they were probably just testing for efficacy. For example, if your proposed compound binds to a certain protein. But as we learn more about other proteins in the body, could the models not also be used to check for at least some other proteins, to check for unintended side effects? Now, I suppose, while modeling has improved a lot with the advent of advances in computing and also software, there's still a long way to go, certainly before you try and have any sort of structure effects. Well, when you're trying to relate to side effects, that's very hard to predict.
I think to an extent, so the medicinal chemists do refer to, or computational chemists do refer to this idea of in silico screening, so screening within silicon chips. And I think there are some common problems that compounds can suffer. So there's some common sort of mechanisms by which the body might degrade or metabolize a compound. And it's possible, I think, to check for that type of thing currently. But yeah, for sure, as we start to build up more genome data and we start to understand things better, then the idea of virtually screening compounds rather than having to actually put them into laboratory-based screening is definitely where the future lies, I'm sure.
Quick definition here. How similar is medicinal chemistry to pharmacology?
They're quite different, actually. So pharmacology is studying the way drugs interact with the body, whereas medicinal, they do of course converge at a kind of a common point. Medicinal chemists are very much chemists who specialize in making compounds. They understand some pharmacology on how the body will process compounds, but they're very much focused on actually making compounds and thinking about what their next target should be based on biological screening results.
And another question here. When testing on animals, many animals have different reactions to the medicines we use. So how do scientists know for definite that the medicine will be effective and not just be a cause for huge concern? So I guess transferring from the animals to humans is the...
Yeah, so that's why clinical trials have to be so extensive. So animal testing is involved, but it's not in any way, shape, or form the be all and end all of this. So a drug can be, I mean, the joke is we can treat every disease in a mouse because mice are always used for this type of thing to start with. But you move from something like a mouse, which is obviously genetically very, very different from a human, and you might find the compound has no effect whatsoever or a very different effect. They tend not to use pigs for animal testing, but apparently the pig is about our closest relative in the animal kingdom, I believe, Dr. Reed. We share a huge amount of our genome with pigs, but no, I mean, animal testing is not really that helpful. And that's why we do student testing instead. So we use phase 1 clinical trials are important.
Excellent. I think some closer in genetic instructions than others. There's another question here. Some clinical trials take so long. What are your opinions on the plausibility of a COVID-19 vaccine being safe?
Gosh, this is testing my testing the boundaries of my knowledge. So obviously, for very obvious reasons, they're accelerating the whole testing program. I think the virus technology they're using to develop the vaccine is very well tried and tested. But sure, there is a potential risk. But I think I would rather take the risk of that vaccine. I mean, things are looking very promising in terms of the trials. They still are doing a program of clinical trials, but I guess the stuff that they have to forego is the potential long-term side effects. But I think these days vaccines it's pretty reliable technology these days. So I would have a lot of confidence in what they come up with. And I'm keeping my fingers crossed.
I think most people are just sort of saying thank you and congratulating each others. But
I've just seen one pop up. Just what course would you do in university to get into that? I would say chemistry. There's some universities who offer degrees in medicinal chemistry as well, but my experience with the pharmaceutical companies are actually, they say, send us your best chemists and we will teach them any aspects of med chem that they need to know. So really, I think chemists are what they're looking for, or other people who have specialized in one science, biologists, pharmacologists. They all have their part to play in that operation.
I think someone asked sort of how long do you study after A-levels to kind of get into the pharmaceutical industry?
So, I mean, typically they would be looking for somebody to have done a 4-year degree. So these days you can do a 3-year degree, which will give you a bachelor's qualification, or a 4-year degree, which most universities offer now, which gives you an integrated master's qualification. And that's usually the level that they want you to go in at. You can then stay on and do further study. So maybe do a PhD, which you're looking at another 3 to 4 years. But we've had plenty of our students who've gone into the pharmaceutical industry in good positions after a 4-year degree. But yeah, you could end up studying for maybe as long as 10 before you do PhD and some postdoctoral work. At which point you're going at a high level, of course.
Sort of relating to careers, a quick question here about how beneficial is a year in industry? Hugely
beneficial. These days, I think there's a big expectation from employers that students will have done some sort of work experience, and that could be vacation work between in the summer break. But if you can actually get paid to do a whole year of your studies, which is the way it works on our program, for example, you can either do the 4-year MSci degree, or you can do the 4-year MSci degree, including a year in industry. So you do a bit of distance learning, but you get paid to do a project in industry, and it's it's not quite, but it's almost like a one-year job interview, really. If you do really well, it will absolutely set your CV alight. And we've certainly had students who've gone on placements who've been offered jobs at the end of them.
And a quick question about enantiomers. So how do you separate enantiomers and test them individually?
Very good question. So to separate enantiomers, you have to do something called resolution. And if you take Prozac, for example, Prozac is an amine. If you want to resolve it, you would form a salt with a chiral carboxylic acid. So there's lots of chiral acids available in nature, like tartaric acid, for example. And if you make a salt with that, then the two different enantiomers will crystallize separately, and you can separate them by crystallization, get rid of the acid, and you've separated the enantiomers.
Can I find any more? Well, I think that's some very good questions. So clearly your talk has sort of resonated with our students, John. So that's excellent.
It's a pleasure.
I think in the interest of time, we'll end the questions there. But thank you once again. Do you have any final comments, Jill, for anyone?
No, just thank you to everybody. Thank you, John. Thank you, Mark, and thank you for all the students who are attending this evening, and we hope you will join us in 2 weeks' time for the next of this series of lectures. Thank you.
Thank you. Bye-bye.
Excellent. Thank you very much, everyone. Goodbye.
Bye.
Hi.
Good night.
So if John and the committee just want to stay on and other people log off and just say a few words. So at its peak, I think we had about 196 people sort of online. So very good turnout.
That's very good, yeah.
So say registered, we reached capacity at 300, but I guess it's easier to register and not just log on than turn up to a lecture theatre. But no, that's an excellent number, a very good start. So went very well. Thank you, John.
Brilliant. Really good.
Well, it has the advantage that I only need to